So our three speakers tonight are Dr. Sarah Boyce, who was a recipient of one of the 2020 Les Muirhoff Botanical Research Awards. Our second speaker will be Dr. Jay Wason, who received the Junior Faculty Award on the floor of New England in 2020. And then we'll also hear from Jacob Suissa, who received the Graduate Student Research Award in 2020. So a really exciting night with multiple presentations, hearing from some of the folks that have been supported by awards from NEBS in the last 2 years. So I'll start off by giving a bit more intro for Dr. Sarah Boyce. Dr. Boyce is the Director of Research and Education with the Linda Loring Foundation on Nantucket. She received her PhD in Ecology and Evolutionary Biology from UConn in 2012 and was an environmental science major at UMass Amherst, so a history in the region. Prior to arriving at the Linda Loring Nature Foundation, she was Conservation Program Director at the Institute for Applied Ecology in Corvallis, Oregon. And while she was at UConn during her PhD, she was also a program coordinator and research assistant for the IPAIN project, the Invasive Plant Atlas of New England. Dr. Boyce has published a number of botanical papers and research on invasive species in journals like the Proceeding of the National Academy of Science, Bioscience, Journal of Applied Ecology, as well as in our own journal, Rhodora. So we're excited to hear from her tonight. She'll be speaking on one of the topic of her Muirhoff Research Award, which is looking at Scotch Broom on Nantucket, a potential invasive in this region. So with that, I'll hand it over to Dr. Boyce. As we've done in other meetings, we'll have, if folks have any questions or comments on Dr. Boyce's talk, you can put those into the chat. At the end of her talk, we'll have sort of an abbreviated couple minutes for a question or two. All right, with that, we will spotlight Dr. Boyce and hear about Scotch Broom on Nantucket.
All right, thank you so much, Jesse. A thumbs up, you guys are seeing my title slide and can hear me. Okay, thanks, guys. So I want to thank everyone for inviting me here today to be part of this this fun talk. I really wish we were doing a potluck. That sounds way more fun than sitting alone in my room. But I want to thank everyone for joining us here too on a Saturday night. As Jesse said, I'm going to be talking about Scotch Broom, specifically on Nantucket Island, and some of the work that was funded through the Les Muirhoff Award. So I just want to thank the New England Botanical Society for not only inviting me, but for the award. The award just definitely helped jumpstart this project, which is something I've been wanting to do as kind of a side project and a pet project for a while. But also, as Jesse said, working with IPAIN as a graduate student, Les Muirhoff was one of my mentors. So it's particularly meaningful to be given this award to work in this field. All right, so because I'm going to be talking about an invasive species, I just thought I would start with an invasion curve. I've given this talk before, and I know this is the Botanical Society, so this might be, you know, kind of everybody already knows this, but it helps to kind of set the stage for when we're talking about Scotch brooms, because I'm going to be proposing this as an invasive species. So when we think of an invasive species or a species novel to a new environment, we think, okay, so if a plant becomes established, over time, it might kind of be in the same area and it becomes established, it's, you know, like low abundance. And then after some threshold, there's rapid population growth, and that for an invasive species. And by the time it reaches this in high abundance over time, and it's noticed as an invasive species by policymakers, by general public, the cost for management is often so high that it's cost prohibitive to manage and eradicate a species. So what we're really interested in is this early time stage, where, you know, there's been a lot of research coming out of UMass Amherst, Bethany Bradley's lab, looking at sleeper species. So which of these species are in this early stage that we can look at and kind of anticipate as a potential invasive. And many of these species that we consider sleeper species, the threshold that they're passing is a favorable climate. So at some point, the climate becomes more suitable for these species and they're sort of awaken and become invasive. And so this is something that I'm really interested in. And this is where I think that Scotch Broom fits in. And I'm going to sort of starting to make the case for that. So my area of interest, as we mentioned, was Nantucket Island, where I'm at the Linda Loring Nature Foundation. It's really nice working on an island when you work on invasive species because you have a very clearly defined boundary. So we're really interested in what is our next invaders, which ones can we deal with and manage for. And our habitats of interest are these sandplain grasslands and heathlands. And I mention them because they have host to a number of rare species, both plants, insect, flora and fauna that are, so these are particularly vulnerable habitats. And so one of the things that we can do when we think about what are our, kind of next threats is looking at similar habitats in other areas. And can we learn from other states and regions? And there's a lot of work looking at, quantitatively looking at areas that have similar habitat and how we can anticipate new invasives. And this is more of a story of anecdotally how I ended up being interested in Scotch Broom. As Jesse mentioned, before I came to Nantucket in 2014, I was at the Institute for Applied Ecology in Oregon. And this is actually a picture of the Oregon coast. It's very similar to, in some habitats, to where I am now in Nantucket. There's coastal grasslands, or called coastal prairies there. There's these interdune swales and really dynamic dune systems, a little more dramatic a landscape than Nantucket, but similar invasive species issues. And so Scotch Broom in this area has become one of the most invasive species in the Pacific Northwest. And when I arrived back on Nantucket, seeing Scotch Broom was concerning. And so that's kind of led to some of this investigation. So before we go too much further, I just wanted to kind of introduce Scotch Broom as a plant. People aren't familiar with it. It's been on the landscape in New England for a very long time, since the late 1800s, mid to late 1800s, as a horticultural plant, dune stabilization, a lot of similar reasons for a lot of invasive species, grows fast and well in nutrient poor sandy soils. So it's a woody legume. It's semi-deciduous, which is quite interesting. The small leaflets do fall off in the fall, but the winged stems of the younger stems are evergreen. So it has a competitive advantage by photosynthesizing virtually all year. It has these signature yellow pea-like flowers, which are very evident in the springtime. So it's really easy to get, volunteers and students out finding these plants. Also great as a plant to look for in the wintertime. And you can see the seed pods there. The seed pods are ballistically seed dispersed. So as the pod dries and it explodes, it can shoot the seeds. The average is like, I think it was 2 to 5 feet from the parent plant, but up to 10 feet from the parent plant. And there's lots of varieties of flower color based in the horticultural industry. So just, it's widely planted throughout the globe, and it's invasive in most of its introduced range, I should say. So in the Pacific Northwest, where it has a long invasion history, it grows, as I said, in these nutrient poor sandy areas. So on the coast and natural habitats like the coastal grasslands, but also roadsides, ruderal areas. It does invade into natural habitats and open space, public lands, forms large monotypic stands, and readily invades disturbed areas like these forestry cuts. So this number that I just put up, $40 million annually in removal and management, is actually an old statistic, but it's for one state, and it's one of the most expensive plants to manage in a place like Oregon, Washington State, up into Canada and British Columbia. And so if a plant is this invasive and problematic, we can kind of learn from these other areas. And it's not just financial, it's the person power in many of these natural areas to remove physically these plants. And so when I kind of went back to the east and I was seeing this plant more and more, and I knew what a problem it was in the West Coast, became interested in kind of what kind of data do we have to see where the species is. And looking at EDMAPS, which is the early detection database, distributional database for invasive species, you can see the counties where it occurs in the United States. So you can see the Pacific Northwest, Northern California, where I mentioned it's highly invasive and recognized as a noxious weed. But when you look on the East Coast, you see basically those coastal communities, it basically follows the Atlantic coastal plain. And so Southern New England, Massachusetts, Long Island, New Jersey, Delaware, you can see a lot of the counties where there's presence of Scotch Broom. And in 2014, when I came back to Nantucket, I was kind of, as I said, I was interested in looking at this. What do we have for data? I was seeing a lot of Scotch Broom on the landscape. And what I was finding on EDMAPS wasn't really reflecting what I actually saw. So there were 4 records, only 4 records for Scotch Broom initially. 2 of them were from the 1900s. So we know that Scotch Broom has been planted on Nantucket for over 100 years. And this is where that sleeper species idea comes into play. The species has been around for a very long time and is only recently being kind of seen more as invasive. I will add that these 1900 records being part of EDMAPS is thanks to Les Meerhoff and the iPain network, which digitized the location information for barium specimens throughout New England. So we have that useful data. The other records were from 2004, which were taken by iPain volunteers when iPain was first started in 2001. And so there are some relatively early records. Also in 2004 is when MIPAG, so the Massachusetts Invasive Plant Advisory Group, first evaluated Scotch Broom as a potential invasive. And at that time, basically, it was determined not to be invasive because of lack of data. So there wasn't data to show that it was spreading rapidly from cultivation. It was proposed because of specific locations where it was known to be a problem. So this photo is from the Provincetown sand dunes, where there were dominant populations, and it was of concern, but it was determined basically that there wasn't enough data. So the MIPAG criteria is longer than this, but these are the 4 kind of specific things I was looking at for where can we add data to. So we know that it's not native to Massachusetts. The potential for rapid and widespread dispersal in minimally managed habitats is quite important. Potential for dispersing away from the site of introduction, and the potential for existing in large numbers away from intensively managed habitats. And so as part of this project, it's really, the Meerhoff Award really allowed us to start this project to be island-wide. So the first step was just mapping all the locations of Scotch Broom throughout the island. And it seems like a pretty straightforward task to let's just document them all. We want to survey specifically minimally managed habitats. So to really, kind of look at that criteria and make sure we're not counting species that people have planted. And we want to document the presence of any reproduction. So one of the thoughts that Scotch Broom hasn't been invasive in the past is that our harsh New England winters have not allowed it to reproduce and have knocked back, if you will, some of the seedlings. And so we want to document what we were finding for reproductive potential. So we used basic open source citizen science programs. So we used EDMAPS, their various apps, and we, as the database that Massachusetts recognizes, we recorded species location using the apps and photo documentation, so very straightforward. And then in addition, we collected evidence of any reproduction that we're seeing, and then note any species of interest that might be impacted. So any rare or endangered species that we were seeing in the vicinity of the recorded locations. We have since learned that MIPAG also looks at iNaturalist records that have been verified as research grades. So that's been very helpful for a lot of volunteers who are really familiar with iNaturalist. So what do we know so far? So I say so far because once again, these awards were awarded in 2020 and we weren't using volunteers for a significant part of 2020. So it was just me collecting data. So we still have a little bit of surveys left to do, but we've made a lot of progress. So we have, say, more than 160 survey points. I need to update this map a little bit, but basically each of these yellow dots is a positive location for Scotch Broom. And each point represents an area, so it could be more than one individual. So this is about closer to 500 individuals at this point that have been recorded throughout the island. And I say it's about 3/4 of the island surveyed, but looking at this next, this kind of blue shaded area, that's the area that we really need to do more intensive surveys on. A few areas have been investigated within that since I made this slide the first time, but we still, I think this winter is going to be finishing up those final surveys. But what do these points actually tell us about what we're seeing for Scotch Broom on the island? So I like to think of this project using Nantucket as a microcosm or as an example of what's happening in other areas. And from a different project, I had already made this vector layer of all the parcels on Nantucket and what their conservation status was. And so overlaying the Scotch Broom points on it, what we can see is that about 74% of the Scotch Broom locations occur in these minimally managed open space. So conserved land, so owned either by the town, state agency, or most likely a private conservation organization. And then there's other non-conserved open space that's usually owned by the town. So a significant amount of these occurrences are in open space. And then I was interested in the types of habitats that these Scotch Broom were invading. And so using the Nature Conservancy's habitat map for Nantucket, which details 37 different habitat types for the island, we found coastal shrublands were the primary habitat. Not surprising, but it kind of begs the question, are the Scotch Broom invading coastal shrub areas? Or if since these are long-lived plants, do they define the habitat that they're invading as shrubland? We see, and so that is further needs to be teased out. We also see pitch pine habitat and grasslands invaded as well. And the grasslands, as I mentioned, are a particular concern because of their importance in conservation. So what does this look like on the landscapes when we go out and we're looking at Scotch Broom in these minimally managed habitats? So this is an example site. This photo is from one of our conservation property on the island. It was an old farm at one point, so the initial population was likely planted, but this is one of the most dense populations now of Scotch Broom on the island. It's an acre maybe of pretty densely populated Scotch Broom. So in this photo, the Scotch Broom is all the kind of spiky plants in the middle ground there. This is a close-up. This photo was taken in early November of last year. So the smaller leaves have fallen off. As it mentioned, it's semi-deciduous. So these are the winged stems that are evergreen. There's lots of dehiscent seed pods still present. And because this is a very old stand, I'm really interested to further investigate the seed bank in this area. It's also particularly concerning because it's on this population that's on the edge of a grassland that does have liatrus and other rare species present. Similarly, this looks like, it's kind of a blur of plants, but the botanists in this group, which is almost everyone, will be picking out individual species, I'm sure. I'm standing on a bike path, and this is looking into a conservation land. So there's, on the right-hand side, there's an adult Scotch Broom in the foreground. And then in the background, these red circles are highlighting some of the individual Scotch Broom that are further into this shrubland. There's mixes of solidago, there's some black cherries in the back, there's huckleberry and bayberry, and lots of other forbs mixed in. So it's a diverse habitat, but the Scotch Broom is invading into this conservation land. We have lots of evidence of reproduction. So we have, obviously the flowers are very obvious and prevalent, lots and lots of seed pods. They can start reproducing as soon as 2 years or 2 to 3 feet big. And they can produce many thousands of seeds and seed pods per individual. They are dispersing over spatial gaps is another thing that we were trying to document. And so there's lots of Scotch Broom along edge habitat, and we're seeing them dehisce across spatial gaps. So looking again at the MIPAG criteria, we know that the species is not native to Massachusetts. we've begun to show that it's definitely in minimally managed habitats, actually primarily in minimally managed habitats. And then we're showing dispersal away from the parent plants. And then there is the potential for existing in large numbers. Of course, large numbers is subjective, so we're trying to just document what we can and reporting out what we're finding. So when I first made this slide for a different talk a while ago, our next steps were to completely map Nantucket and then start the process of proposing to MyPAG. But then in the meantime, the representatives from MyPAG, which again is the Massachusetts Invasive Plant Advisory Group, asked us to put together a proposal for Scotch Broom. And so in September, we presented our findings of Nantucket and other areas in Massachusetts and proposed listing of Scotch Broom. In December, Scotch Broom was voted as likely invasive for Massachusetts. And so the next step with that would be for the MyPAG ruling to be taken up by MDAR to consider it for state listing. And once it's on a state list, it would be considered banned from the horticultural trade and not be able to be planted or transported. But for us on Nantucket and those of us managing these landscapes, it's a first step in helping us kind of justify the continued management, gives us some teeth when we talk to people about not planting it further. And it's really interesting as we're thinking about our changing climate, how the species will continue potentially to spread. And I just want to mention really quickly, we were proposing the listing as for an invasive, and it was listed as likely invasive. And the difference with there is really in this one criteria, and this is a lot of discussion came up, and I mentioned it specifically to this group because we're a bunch of botanists. Is it widespread in Massachusetts, or at least common in certain habitat types in the state? And so there was a lot of discussion about this. As I mentioned, when we first looked at the EDMAPS map, Scotch Broom is really following that coastal plain habitat type, those soil types of the coastal plain. And as we see, it's really common in Nantucket, Martha's Vineyard, Cape Cod, Plymouth County. That's where we're seeing more evidence of invasive potential. But because it's not invasive in other habitats within the state, it's not considered invasive for the potential, not as a full invasive for the whole state. But a lot of it is just us making observations and recording it. So especially for this group, something I'm interested in is not just for Scotch Broom, but for a lot of new species, established species that might be becoming more invasive or having more invasive tendencies, to record them, whether it's iNaturalist or EDMAPS. These data are really important as people are investigating, as species ranges might be changing and things might be, sleeper species might be becoming more abundant. Having the data is more important. We're learning, obviously data is important, but we're learning as we're trying to propose some of these species, the importance of data from all throughout the region. So that's my plug for please record your data, not just in your notebook, but get it out there and share it with others. And I think with that, I'm happy to take a few questions if I have time.
Thank you. That was fantastic. We're going to take a question or two from the comments section. One or two have come in now. Alice had asked if this is a species that appears to persist as forest redevelops during succession. Any thoughts on sort of its successional status, whether it persists as habitats shift towards forest?
Right. So I know that's definitely true for things like Japanese barberry, that come in and then persist as a secondary, forest grows in. I think for Scotch Broom, that has been seen to some extent, but it does really like the light more. So it prefers those edge habitats. It does really well. On Nantucket, we don't have the forest issue, but I know that is something that's seen more on the West Coast. And it's, it's affecting those kind of clear cut areas of the forestry trade, but it's not as not as much as like Japanese barberry that will like persist in the understory.
Yeah. All right. We also had a comment in the chat section from Lisa Stanley, and this is a wonderful point too. In addition to sort of the digital documentation and iNaturalist and EdMaps, she was suggesting that people should also be collecting herbarium specimens to create these physical vouchers of its location and sort of status at this point in time for future reference in herbaria. And then Bob asked if it's still being sold in nurseries on Nantucket and other places along the coast.
Yeah, our local Stop and Shop sells it every year. Yeah, so it's so that's, you know, I've given this talk, a similar talk before. People always ask, what's the point of listing it if you already know if it's a non-native, then you can just manage it from your habitat. It's like, yes, for the land that I manage as a professional, that's true. But for things like talking to the distributors, the landscapers and homeowners and things about planting it, having that listing is actually kind of gives you a little teeth to make that argument. And so it's useful for those purposes too.
All right, and then we'll just do one more comment, and then if people have other questions, we can certainly, we'll have an open period at the end of the overall meeting here. But Mary had asked or commented, wondering if there's historical or observational data on the species going back through the 20th century, maybe to the late 19th century. Her impression was that the species might be less prevalent on the island now than it was decades ago.
So I think the popularity piece is one of those horticultural popularity things that where you see it a lot and then it kind of waxes and wanes with whatever's popular, especially in a place like Nantucket where people landscape to the nth degree. There is, there's historic records, as we say, for the herbarium specimens. And there's, like writings that we know about from different areas of the island specifically. But I know some of it was brought in because it's beautiful with the flowers, but then also that soil stabilization is why it was brought into like Cape Cod and Nantucket and the Vineyard and places like that. Great.
All right, thank you. That was a wonderful presentation to kick us off tonight. So our second speaker tonight is Jay Wayson, who is the winner of the 2020 Junior Faculty Award on the floor of New England. This is one of our newer awards that just started up 3 years ago, targeted to new faculty who are setting up shop, setting up research labs, focused on botanical topics in the New England region. Jay was our second winner of that award. He is an assistant professor of forest ecosystem physiology at the University of Maine and the School of Forest Resources. He received his PhD from SUNY ESF, the College of Environmental Science and Forestry, in 2016 out in Syracuse. He also holds a Bachelor of Arts degree in Environmental Studies from the University of Pittsburgh. His research focuses at sort of at the interface of tree physiology and ecosystem processes with consideration of environmental change factors like acid deposition and climate change on forests and tree species of the northeastern US. He's published his research in a range of journals, including the Annals of Botany, Annual Review of Plant Biology, New Phytologist, the Northeastern Naturalist, as well as the American Journal of Botany. Tonight, he's going to be speaking on some of the research that was supported in part by the Junior Faculty Award back in 2020, investigating drought effects on tree physiology, growth, and survival for some northeastern tree species. So with that, I will turn it over to Jay to tell us about his research on tree physiology.
Great, thank you, Jesse. Appreciate the introduction. Thanks everyone for being here tonight and inviting me to speak today, and thanks to the New England Botanical Club for helping to fund some of this research. So tonight I'm going to be talking to you about an experimental research project that we did here at the University of Maine up in Orono, where I've been an assistant professor here now for about 4 years. And as many of you know, any good project needs to have a good acronym. It took us a long time to come up with this one, but this is the Drought Time Project, and it stands for Drought Timing Impacts in Maine. That's the ME at the end. That one took us a while. And so today we're going to be talking about how the timing of drought impacts the physiology, growth, and survival of trees. I'm going to be focusing mostly on the growth just to keep things on track tonight. So as many of you are aware, drought is relatively rare in the northeastern United States, especially in parts of the region where I often work. So this is a picture of a spruce fir forest where I've done a bunch of research as well. And oftentimes we go for walks and this is what we walk in. And so when I tell people that I focus on drought and the impacts that drought has on the physiology of trees, they're often surprised that it's something that they should be concerned about in the northeastern United States. But what we consistently find when we study trees in our region is that they're often still limited by moisture availability. One way that we can study this is using the tree ring record. And so this is a relatively complicated graph that you don't need to focus the details on, but this is from a recent publication that looked at black spruce increment cores, so growth rates of trees over about a 100-year period. And this is in southeastern Canada. And here we're looking at the relationship between growth and in the center column, precipitation. And when these circles are red, that's suggesting that increasing precipitation leads to decreasing growth. But when those circles show up blue, that suggests that increasing precipitation leads to more growth. And so here what we can see is a really strong, consistent effect of a lot of moisture in the prior year summer leads to increased growth the following year. So that suggests that oftentimes these trees are limited by moisture. And this is especially important as we move into the future because we're expecting climate change to increase the likelihood of drought. The other important aspect of drought on trees in our region, and these data do come from our region too, is that the impact of drought doesn't just affect trees in one year, it often has this legacy effect like we just saw in the last slide. And so the graphic on the left here is from a recent publication looking at the effect of drought. You can just think of generally just how much does it affect growth on the y-axis, and then the different months of when the drought actually hits. Here, the white circles represent the drought effect of the current year, and you can see droughts that occur in May, June, or July have the most impact on growth. This is across a wide range of tree species. But you can also see these black circles here, which represent the legacy effect of drought. This is the impact of drought in the subsequent year, so it causes long-term reductions in growth. And what you see is that droughts, especially that happen later in the summer, so a drought in August, for example, has a larger effect on growth the following year than it does on growth in the current year. But what we haven't been able to figure out with this type of study is what are the actual physiological mechanisms, or how are the plants actually responding to the droughts in those time periods? Because the most effective and efficient way to have lots of data on this is to look at increment cores. And this is becoming especially important. This graphic on the right is just representing how in the future, as we move into near the end of this coming century, we're expecting climatic water deficit to become much reduced from where it currently is today. So this is going to become more and more of an issue. These drought effects and legacy effects are likely to persist in our forests. So for this study, what we decided to focus in on is the smaller size classes of trees. We know that small trees can be especially sensitive to large-scale environmental changes. And we have very little data on how regeneration responds. And so, for example, this little spruce tree hanging out on a mountain right now, how does that tree actually respond to these droughts that are occurring? One of the things that's also rather difficult to pull out from tree rings is actually something that's very important for small trees, which is height growth. And so in this study, we also really emphasized the impacts of drought on height growth, which allowed us to better understand how drought might change species competitive dynamics in the future. So the overall objectives of this study were threefold. The first is to better understand the phenology, or the timing of height and diameter growth for sapling trees of 6 tree species from our region. Next, we wanted to focus in on how droughts impacted height and diameter growth during different seasons. So we were going to implement droughts at different times of the year to see how it impacted those 2 aspects of growth. And finally, we wanted to tease in on this interesting idea of intra-annual recovery. So that being, if there's a drought in one year, in one part of the year, but then the drought ends later in that summer, for example, can the trees actually recover and have some resilience to that drought? And that becomes especially interesting because that doesn't necessarily get preserved in the total growth of the tree in that individual year. So this is where I do a quiz. I almost always do a quiz in any one of my talks. We don't really have a good opportunity to chat to each other today, so you can just kind of think to yourself. I told you I work with 6 tree species. This is a picture of one of our studies. So you can take a minute for yourself and see if you can identify the 6. I kind of pointed out a couple of spots where you can look in here. So number 1 down here, I'm pointing at this kind of curled leaf. It's kind of actually part of this partially dead stem here. And that one is red maple. So one of the species we focused in on was red maple. Number 2 is up here. It's this really large one. It kind of dominates the background as you look across this. So you're starting to get the sense it's a relatively fast growing tree. And those of you looking at the serrations on the leaves would pick up that's probably paper birch. So we did include paper birch in this study. I tried to include a picture here that had all 6 species, and I struggled to find one of them. I think I picked it out back here. It's this kind of elongated leaf here that's got a slight red tinge to it. That's about all you can see of the plant. I imagine some of you might have a guess for it, but that one's black cherries. We included black cherry in this study. And then the rest are a little bit easier to see. I just love the color of the foliage on number 4 here. So that's our eastern red cedar. Number 5 is represented by this pine. So that's a 5-needle pine, Pinus strobus. And then finally, number 6, hanging out on the bottom here, which is a favorite tree species of mine, is northern white cedar. So those are the 6 species we chose to work with in this study. They represent angiosperms and gymnosperms and kind of a range of different potential future success in climate change scenarios. So when I first started here at the University of Maine, my first summer here, we built this hoop house, this experimental greenhouse we affectionately call the Drought House, and we planted 288 individuals of all 6 of these species, each one in its own individual 5-gallon Lowe's bucket with holes drilled in the bottom, and they were each irrigated individually. And so this is kind of the experimental layout of our treatment design. We had 12 experimental blocks, and within each of those blocks, we had 4 individuals of each of the 6 species. Here's an overhead picture of what one looked like. The trees were allowed to grow for 1 growing season. So most of them were about 2 years old when we planted them as bare rootstock. They grew for 1 additional year. And then in 2020, we initiated our drought treatments. And so there were 4 different treatments that we initiated. One was just an irrigated control where the plant got enough water. One was a spring drought, one was a summer drought, and one was a kind of late summer fall drought. And so then we were going to track growth and physiology over the course of those droughts. And we imposed those droughts just simply by putting a canopy on top of this structure and leaving the sides open so that we didn't increase the heat too much inside there. So without getting into all the details, essentially what we did here is weekly measurements of physiology, soil moisture, height, and diameter growth. I'm mostly going to focus on the soil moisture, height, and diameter results today. And when I say we, I primarily actually mean Ruth. This is Ruth, my graduate student. This was the focus of their master's research projects. And so what did we find? So I'm going to introduce you these graphics that have a bunch of colors on them. And they're going to demonstrate the different drought treatments that we actually imposed here. So on the x-axis in this graph, we actually have day of drought. And so 0 representing the day right when the last day that those trees got water, and then the time period after that where they were didn't have water. The droughts lasted for 6 weeks in each of these scenarios. And the y-axis in this case is just soil moisture. And for reference, we have some physiological data that gives us some reference for this as well, but this dashed line at about 5% is really when you start to hit stressful conditions. And generally what we found, especially from the physiology, is that we only imposed a relatively moderate drought on most of our trees, which ended up making this study really interesting. So each of these panels represents one of the 6 species, and this is how they'll be ordered in the next 2 slides as well. And then within each of those panels, we have the spring, summer, and fall drought as different symbols and colors. And then along the top here are the controls. And so those are the ones that were continuously irrigated. So you can see overall, the broad pattern is that the spring droughts, the soil moisture did not decline as quickly as it did in the summer and fall. We think part of that is because the spring was not as hot and the air was not as dry, but also the plants were mostly smaller that time of year. They didn't have as many leaves, and so they weren't transpiring as much water. One of the interesting things you see right away is paper birch. So paper birch within just about a week used almost all of its water, and you'll see that most of the trees die within about a week of implementing the drought. Even the control trees, regardless of giving them, I forgot, like 1.5 gallons of water every night, they still were drawing down their water every single day. So paper birch, they were very large, but also high water users. And so with this broad context, here we imposed a moderate drought. Most of our trees actually survived the drought, and paper birch is one of the only ones that really died throughout the process. So let's take a look at how these droughts impacted height. These graphs are similar, but they're also a little bit different. So I'll draw your attention to the x-axis here, which now is showing day of year instead of day of drought. So here we're going from early June all the way through to the end of August and early September. And so this is the entire growing season for those plants. And on the y-axis, we're demonstrating relative height growth here, which is just height relative to its start at the beginning of the spring drought. Those colored bars along the bottom indicate when they were receiving either the spring in pink, summer in yellow, or fall drought in that dark green color. And now each of these lines then represents the average height growth of all of the trees of that species. So again, we have our 6 species in the 6 panels. The controls are in the blue triangles here, and then the springs are in the pink squares. And so one of the things you can immediately see, especially if you look here at red maple, is we do have an impact on height growth. You can see height growth of red maple levels off when it's experiencing this spring drought. But then after we rewater those trees in the summertime, they're actually able to recover. And the statistics told us at least that they were not significantly shorter than the red maple trees that received water that entire year. You can also see that for paper birch, we experienced a lot of mortality. So you can see immediately the height growth in the spring trees stopped and none of them lived. So that's the end of when we had any alive. Similar thing for the summer drought for paper birch, but you can see they achieved much more of their growth before that drought hit. I'll draw your attention to white pine here, Pinus strobus. You can see white pine has a phenology pattern where it does most of its height growth very, very early in the spring. So that's the blue line here. So that's just the well-watered trees, and they don't do any more height growth later in the year. So you wouldn't expect a drought later in the year to have any impact. And sure enough, what we see is that the spring drought does cause a reduction in height growth for white pine, but later in the summer and fall, it does not result in a reduction in height growth. And finally, I'll draw your attention to northern white cedar in the bottom right here. This is a species that I find really interesting. It showed really strong resilience in both height and diameter growth. You can see it continues height growth throughout most of the growing season, the blue line there. And the spring drought, you can see that we reduced growth, but this species showed strong resilience and in the end recovered, and the growth of the spring drought trees was no less than the trees that were watered the entire year. Interestingly, though, the summer drought in yellow actually did truncate growth and cut off growth, and they did not have the ability to recover and come back. So for a species like northern white cedar, a summer drought is actually really detrimental to their height growth. And so this is important, we think, for competitive dynamics of species interacting in the forest. The story was similar for diameter, but a little bit different in some ways. Diameter was a little bit more variable, in part because the resolution of our measurements and the precision of our instruments. So you can see we have quite a bit of variability. This is where I'll also tell you that red maple and black cherry, we did experience some frost damage. And so we did have some issues with survival in those. And that's why we have kind of a big scatter plot for red maple and black cherry in this case. But again, you can see paper birch, you can see that mortality. You can see this interesting effect for the paper birch that they shrink. And so any of you that study this kind of thing, you recognize that the trees, when they're fully hydrated, they swell up. And as they use their water, the stem of the tree will actually shrink. And we actually were able to capture that just using simple calipers in this study. And then again, I'll just draw your attention to northern white cedar that experienced a decline and actually shrinkage from that spring drought. But as soon as we rewatered that tree, it was able to, those trees, they were able to recover and achieve the same total growth at the end of the growing season. So now that we've seen how drought actually impacts height and diameter growth of these trees, let's think about why. What is the physiology behind this? So the reason that trees reduce their growth during or after these drought conditions is because they close their stomata. So I'm sure that this is familiar to most of you. But the stomata are the pores in the bottoms of the leaves that allow gas exchange with the atmosphere. And so in order for the leaves to get the carbon dioxide they need to fix photosynthate and produce the sugars that they need to grow, they have to have these stomata open. It's like having little tiny mouths open on the bottom of the leaves. But in the process of doing that, lots of water evaporates out of those leaves as well. About 400 molecules of water leaves the leaf for every 1 molecule of carbon dioxide they bring in. So this is a really expensive trade-off. So the plants close their stomata when there's not enough water, and that causes them not to be able to bring in any carbon dioxide. So that's the mechanism by which they have less growth. Interestingly, we also find some other research we're doing in our lab, and others find this as well, is that dry air can actually have a similar effect on these trees. And so this is a graph of photosynthesis on the y-axis relative to time of day from the morning all the way through to the afternoon. For red maple, these are for relatively large red maple trees that had plenty of soil moisture. It rained just a few days before this, but you can see photosynthesis around the middle of the day drops to about 0. That's because it's at the hottest point in the day and the air feels the driest to those trees, and they can't transport enough water. And so this is another research area that we explore in my lab as well. It's kind of contrasting these soil droughts to these atmospheric droughts that are relatively short-lived, but can have a really strong impact on the ability of the plants to sequester carbon. So what are the implications of this greenhouse study that we did? What we found is that tree responses to seasonal droughts are dynamic at these intra-annual timescales. We detected things with the growth rates of our trees that you would not see if you were just looking at increment cores of long-term growth patterns. So we found that this total annual growth is not necessarily capturing the resilience of some of these trees. And so we're improving our understanding of the resilience of these trees to these novel experimental, or to these novel climate conditions that we expect to be more important in the future for these trees. This is really important because we haven't exposed the trees in our landscape to these conditions, so we don't know how they're going to respond necessarily to future climate. We also find that these drought patterns likely drive these one-year lags that are really, really common for tree growth responses to drought. So even though the trees in our study were often able to be resilient and recover their total growth that year, we ended up harvesting all the trees at the end of that year for a separate related study. But if we had left them growing for another year, I bet we would see that they had their total growth was lower in the following year because they had not sequestered as much carbon in the year when they received their drought because the stomata were close. And just a little plug for some of the other work that we do in our lab. We have a range of different projects, but another one you might be interested in is a study that's looking at how droughts and floods reduce survival for saplings and seedlings. And so these survival studies require a huge sample size of lots of trees, so you can kind of do this dose response approach. And we're doing this for balsam fir and northern white cedar, which are two species that compete in these northern white cedar swamps in Maine. So I just want to make a quick acknowledgement to Ruth. This was their master's thesis and it was a ton of work, and they did a great job on this. Nick Fisicelli from the Scudic Institute and John Zhang here at the School of Biology and Ecology at the University of Maine were integral in this project, and all the folks that helped out with all the lab work here and the field work. The New England Botanical Club that funded some of the tools that we use for the physiological portion of this research that I didn't end up reporting on today, and the Maine Economic Improvement Fund and the USDA for funding this work as well. Thank you. I'll take any questions.
I guess to lead off, I'm curious to know if you see connections between the work you're doing here and this idea in ecology about sort of like the seedling recruitment niche of species, and whether you might actually see differences in the climate tolerances of these early life stages compared to well-established adults, and whether populations of trees may be eventually affected by that, where the adults may be surviving, potentially suffering from these effects, but recruitment may disappear or drop off in populations.
Yeah, that's a great question, Jesse. It's something we talk about all the time. And it's kind of this big question as our northeastern forests and plants more broadly move into these novel climate change conditions. We don't really necessarily know which parts of their life are most sensitive. And so one of the ways that we approach that is using the kind of exposure and sensitivity framework for thinking about how exposed are these plants to those conditions, how sensitive are they kind of physiologically, that's where the physiology comes in, and then how does that relate to their risk. And so we actually have some projects going on now. We can think about exposure for sapling trees, for example. In the understory of the forest, they're not necessarily exposed to the hot and dry conditions as much. So maybe they're buffered a little bit from the canopy from some of those climate change effects, whereas the canopy is more exposed to some of those things. However, the large trees may be more resilient to some of that and less sensitive. So that's definitely where we want this work to go. And I think, yeah, we've got some ideas for some projects that help to kind of bridge that gap, because that's where I think we really have a lot of learning to do on how climate change is going to impact the demographics and for these species.
Yeah, and it's a tricky issue too, because if you're looking at natural populations and some event like this had happened 10 years ago, 20 years ago, they would just be missing. Right. If you saw the type of mortality you saw. So there wouldn't be tree cores there to take a reading off of. The trees are just not there, or the seedling stage would not be there. So
yeah, that's the big problem for tree ring studies, is you don't know.
Yes. Yeah. All right, our third speaker tonight is Jacob Suisa. Jacob was one of the 2020 NEBC Graduate Student Research Award winners. He is a PhD candidate in organismic and evolutionary biology at Harvard University, where he works with Dr. Ned Friedman, the director of the Arnold Arboretum. Prior to starting the PhD program at Harvard, Jacob received his Bachelor of Science degree in plant biology at UVM up in Vermont. And between his undergrad work in plant biology and starting at Harvard, he was an independent research fellow at the Smithsonian's National Museum of Natural History, where he worked on a project related to the evolution of Izoides. He has an impressive track record of publications just in the last few years, since 2018. He's published 9 research papers, 8 of which were first authored in journals like the New Phytologist, Annals of Botany, American Fern Journal, and Molecular Phylogenetics and Evolution. So a really exciting trajectory of a research career in botany and focusing on ferns and pteridophytes. But then even beyond his academic work, Jacob is a really standout example of a very successful science communicator, spreading sort of the appreciation of plants and botany to a very broad audience. He has a large following on social media, including a popular Instagram series called Let's Botanize, which is definitely worth checking out if you want to venture into social media. Tonight, he'll be speaking on a topic related to his Graduate Student Research Award from 2020, focused on the evolution of the fern vascular system. So we'll let Jacob take it from there.
Great. Thanks so much, Jesse. I really appreciate that. All right. So first, I want to thank NEBC very much for inviting me to speak about my work today, and you all for coming and staying this late. So I actually won't be talking about the project that was funded by NEBC, but rather something else that is a bit more complete, but definitely tangential. And I'm very excited to be presenting some of the findings from this project, which focuses on the evolution of the fern vascular system. And today's story really centers around plant structure, development, and evolution, and how these processes have played out across deep time. But before we get there, I'll start with explaining what primary vasculature is and why it's important to study. So when we think of plant vasculature or plant vascular tissues, usually the first thing that comes to mind is wood or secondary xylem. However, this is just one half of the full story. So every vascular plant starts off their life with primary vascular tissues, which you can see here on the right, dispersed in these vascular bundles. And as many of you know, these primary vascular tissues are the original tissues produced by the growing point of the plant. And many plants like ferns, lycophytes, monocots, and many other herbaceous eudicots never produce secondary tissues or wood, meaning that they rely entirely on their primary vascular tissues for survival. So what do these vascular tissues look like in a fern? Well, if we cut open a fern, like you see in this center picture, you'll see some of these white tissues, and these white tissues are the vascular tissues. And if we take a thinner section and we zoom in under a compound microscope, you will see a variety of cells forming particular tissues. Of interest are these tissues that are concentrated in the center that are fluorescing in purple. And these are the tracheary elements, which form the xylem or the water conducting tissue. And flanking either side of the xylem are the cells that form the phloem or the sugar conducting tissue. However, as we all know, plants did not always have these vascular tissues. And vasculature, particularly the xylem, evolved around 425 million years ago. And if we went back in time to the point where vascular tissues first evolved, the terrestrial world would look very unfamiliar, with many vascular plants scattered around a barren landscape, like you see in this photo. However, this early unfamiliar landscape of the Silurian and the Devonian is really juxtaposed by the both familiar yet so foreign landscape of the Carboniferous period, only 50 million years or so later. And by this time, vascular plants, particularly the ferns and their relatives, radiated to cover the earth in a dazzling display of green across the landscape. And what really allowed these plants to do this was the thing that you cannot see in this photo. And that is the evolution and the diversification of the internal vascular system. And the evolution of this proper vascular system was one of the most important innovations in land plant evolution, because these novel tissues really amplified mass flow rates by orders of magnitude, allowing plants to increase their photosynthetic capacity and grow much larger. And this led to dramatic changes in the terrestrial landscape and atmosphere, including carbon dioxide sequestration and increased oxygenation, in turn profoundly affecting the course of evolution for life on land. And when we look across living vascular plants today, we see some of the most striking displays of primary vascular patterning in the ferns. And this diversity naturally leads us to ask two major questions. Why does this variation exist? And how has this variation evolved? And broadly speaking, my work has explored these questions by integrating methods that lie at the intersection of structure, function, and evolution. But today, I will just focus on the evolutionary aspects of my work. So specifically, I will aim to explore how variation in the construction of the vascular system evolved across fern macroevolutionary history. So what do we know about fern vascular architecture, or fern vasculature? Well, the study of fern vasculature is not new. It's been a topic of intense botanical review for the last 150 years or so. And much of this early work focused on understanding the structure and really categorizing the different types of primary vascular architectures, some of which you can see outlined here. And over time, researchers began to develop hypotheses on the sequence of events in the evolution of vascular architecture in ferns. And eventually, consensus emerged around the notion that vascular architecture evolved through a largely unidirectional and stepwise increase in complexity, from simple to these more complex architectures in a stepwise ordered progressive sequence. However, none of these long-held hypotheses have ever been examined with modern comparative phylogenetic methodologies. So many questions remain unknown. Specifically, we do not know how many vascular architectures even exist, which model of vascular evolution is most likely, what drives major changes of architectural patterns, and how rates of vascular evolution have shifted throughout fern macroevolutionary history. And these are the questions that I aim to answer today. So in order to answer these questions, I first needed to assemble anatomical data on stem vasculature for thousands of fern species. Specifically, with the help from my amazing undergrads, Skyla and Paul, I was able to generate information of fern vascular architecture for 3,339 species, or 32% of all fern species, spanning 85% of fern genera, 96% of fern families, and all of the fern orders. I was also able to gather information for 12 extinct fern species from the fossil record to morphologically calibrate the phylogeny. And I'm happy to talk about what that means at the end if you're interested. So I combined these data with an existing fern phylogeny to answer my questions. Now, I use some pretty complex methods, and I'm just going to skim over them. But if you have any questions at the end, I'm happy to answer them about those. So I first delineated vascular architecture using quantitative image analysis and phylogenetic principal components, as well as hierarchical clustering. Using this circumscription and a robust phylogeny, I then tested 18 models of character evolution and then reconstructed the evolution of vascular architecture. Then, to determine the underlying drivers of major structural innovations in vascular architecture, I explored the relationship between climate as well as body size, particularly stem and leaf size, and major character transitions in vascular architecture. Finally, to examine how rates of phenotypic evolution of vascular architecture are associated with major historical fern radiations, I explicitly investigated temporal shifts in rates of vascular evolution and lineage diversification. So before I could explore these broader questions about macroevolution and these patterns, I first had to ask if variously circumscribed architectures were supported quantitatively. And this was important because there have been classification systems, over the last 150 years that group as little as 2 vascular architectures and as many as 20. So I really needed to test this quantitatively. And what I found was that there are essentially 5 quantitatively supported distinct vascular architectures. And of course, the distinction between these forms may merge together in certain parts of this morphospace that you're looking at here. And this may reflect the natural variation between the different architectures. Also important to note for downstream results is that I found one polymorphic form between this purple architecture, the selenostele, and the brown architecture, the dictyostele, which just means that in some species, these 2 architectures can occur in the same individual stem. So using this framework, I then explored how vascular architecture evolved across fern macroevolutionary history. And I first revealed that these long-standing canonical theories on the stepwise evolution of vascular architecture were actually too simplistic to explain the evolution of vascular architecture across fern macroevolutionary history. Rather, a new theory that accounts for increased number of transitions, rate heterogeneity, and multiple evolutionary reversions was much more likely. And this was really important because it fundamentally shifted our understanding of the developmental steps in the evolution of vascular architecture. So next, I needed to address why these transitions occurred. So for more than a century, botanists have speculated on the functional correlates of vascular architecture. For instance, some scholars propose that certain architectures correspond with increased rates of water movement or mechanical strengths of the stem. More recently, physiological work on the hydraulics of fern leaves and seed plant stems has suggested that increased vascular dissection actually confers an increased resistance to drought-induced hydraulic mortality. And if increased vascular dissection is hypothesized to increase drought resistance, it may be expected that fern species with highly dissected vascular architectures should occur more frequently in ecosystems with a high potential for drought. And this is a trend found in woody angiosperm shrubs. In fact, we did not find support for this hypothesis, as there was no significant relationship between vascular dissection and mean annual precipitation, as you can see here on the right, meaning that any vascular architecture can occur in a variety of different rainfall regimes. Furthermore, in our prior research explicitly investigating the physiological implications of vascular architecture in fern stems, we did not find evidence that increasing dissection leads to an increase in drought tolerance, further corroborating these findings. Instead, we found that major architectural changes are actually driven by shifts in stem size. So what you can see here is that increases in stem size are strongly correlated with the development of a pith. In other words, as stems grow in diameter over evolutionary time, they eventually reach a critical threshold, interestingly around 5 millimeters. And then we start to see species develop a pith. And this is really important because it showed us that major changes in vascular architecture are driven by shifts in body size. Moreover, this specific transition from the protostele to the selenostele, or from lacking a pith to having a pith, quite literally sparked the diversification of vascular architecture across ferns, because from the selenostele, transitions to almost all other vascular architectures are possible. So finally, we needed to understand how these evolutionary processes have played out across deep time. And to do that, I explored how rates of vascular evolution have shifted across the 400 million years of fern macroevolutionary history. So how did I do this? Well, imagine you have a trait, and across a lineage of organisms, that trait evolves under a certain rate of change. Let's say 2 changes every million years. And if we color the branches based on their rate of morphological evolution, it may look something like this, where every branch is evolving under the same rate category. However, we could imagine a scenario where the same trait could evolve under a different rate of change across different lineages. So for instance, there could be some lineages where the trait evolves very fast. Let's call that the red lineage. And we can have another part of the phylogeny, another lineage, that is evolving under a very slow rate of change. Let's call that the blue lineage. So this is exactly what we did. And using recent phylogenetic comparative methods, we were able to determine that rates of vascular evolution fall under two rate regimes across fern macroevolutionary history. We have rate 1, or the slow rate, with equal probability of transition to all vascular architectures, and rate 2, or the fast rate, with varying transitions between all architectures. And what you can see is that across the fern phylogeny, most branches fall under rate category 1, or the slow rate, while early branches and some clades fall under rate category 2, or the fast rate. And when we place these rates into the broader context of evolutionary time, we gain some deeper insights. So when we plot the rate of vascular evolution as a function of time, as you can see by focusing on this purple line, we find that these early fast rates of vascular evolution occurred during the Carboniferous period. And this is important because based on the fossil record, we know that ferns had a major diversification event during the Carboniferous, and these high rates of vascular evolution seem to correspond with this event. In contrast, during the Cretaceous, rates of phenotypic innovation of vascular architecture were very low. And this is interesting because based on molecular phylogenies, we know that ferns had another rapid diversification event in the Cretaceous. But during this time, rates of vascular evolution seem to be very low and perhaps decoupled from speciation. In other words, rapid phenotypic evolution underlies early, but not late, fern diversification. So overall, if we return to our main questions, we can come away with 4 main points that reveal novel insights on fern macroevolutionary history and the evolution of vascular architecture. First, we can say that there are 5 quantitatively supported vascular architectures across ferns with 1 polymorphism. The second is that old stepwise unidirectional theories on the evolution of vascular architecture were too simplistic. And a new, more complex theory of vascular evolution is much more likely. Third, we can say that major shifts in vascular architecture are not due to physiological or ecological, rather hydraulic demands, but rather are due to shifts in body size, specifically stem and leaf size. Lastly, we can say that there are temporal patterns in the evolution of fern vascular architecture, specifically that the rapid diversification of vascular architecture accompanies the early diversification of crown group ferns, whereas during the more recent Cretaceous diversification, rates of vascular evolution were low and seemingly decoupled from cladogenesis. So, as Jesse alluded to in the beginning, I wanted to end my talk with talking about some of the science communication work that I've been doing with my colleague and amazing friend, Ben Goulet-Scott. So for over a year now, we started this Instagram and YouTube-based account called Let's Botanize, where we wanted to share our passion for plants with people around the world. And we realized that our small project really had a much larger purpose, and we really leaned into that. This broader purpose is, we think, to democratize the study of plant biology, which we believe will enrich people's lives and hopefully allow us as a society to become better stewards of the earth in light of the climate and biodiversity crisis. So this is all to say that I think my research is very important, but it's just one dimension of who I am as a scientist and a person. And if you all are interested, I highly recommend following us. You can follow us on YouTube or Instagram, and we're even on TikTok if you're on that as well. So with that, I'd like to thank you all for coming, and the NEBC for funding parts of my doctoral work. And now I'll take any questions if there are any.
All right, Jacob, thank you very much. A fascinating talk on fern evolution, and then also just, again, sort of congratulations on the science communication work you're doing as well. That's a great thing for the community, and bringing some of this sort of joy of plants out to the broader society. So questions can be popped into the chat here. We've got one or two coming in. Bob is asking about whether there's any evidence of horizontal gene transfer during the evolution of ferns. Any thoughts there?
Yeah, I haven't worked on that particularly, but definitely some of the folks who do are, Fei Wei Li at Cornell. He's done some work on that, and actually Charles Davis at Harvard, who's a professor here, has published a bit on, I think, one paper on that as well. So the answer is yes, but that's not my area of study, so I can't really speak too much about that.
Right, and then another question about whether, if you're thinking about the fern flora, the pteridophyte flora of our region of New England, are there any species that stand out as having unusual architecture or particularly striking examples of some of these different forms that you've described? Or is this the sort of thing that you see mostly in the tropics for some of the diversity of forms?
No, for sure. We have, a rich fern flora here for sure. And we do have probably I'd say almost all representatives of some of these architectures. So, the bracken fern, if you cut open those, if you can get to those deep rhizomes, as some of you know, they have these beautiful, polycyclic architectures. And we also have, the Virginia fern, Polypodium virginianum, that has one of these beautiful perforated dictiosteels. They look sort of like U steels. They'll look like an herbaceous U dicot if you cut them open. There are some of these more complex architectures that occur in the tree ferns, which are, of course, restricted to more tropical regions. But yeah, we have some pretty nice diversity in vascular architecture up here for sure.
All right, and then we had a question in from Kathy, wondering if you could say a bit more about this relationship between plant size and vascular complexity.
Yeah.
Sort of know it's a thing, but what might explain it? Is there any mechanism there behind that shift associated with body size?
Yeah, absolutely. So one thing that I didn't say or didn't mention, which Kathy definitely knows, is that all ferns start their if they are they start their life as with a protostele when the spore phyte is very young, when the rhizome diameter is small. And this is all work that's been done, about 120 years ago now, by Tansley and folks like that. But and as if it's not protostelic at maturity, as the stem diameter increases, a pith will develop, and that's developmental within an individual. We actually were able to recapitulate that looking across species, across the phylogeny. So that also stands at a macroevolutionary scale. The developmental, and that's, I will also say, this is something that's also found in the fossil record with some lycophytes, as well as seed plants, in particular with roots. And there actually was a really nice paper that just came out, I think it was AJB, looking at pith diameter and twig diameter, and there was a nice correlation there as well. But I kind of am going off topic here. To answer Kathy's question, in plants with primary vascular architecture, the mechanism there essentially has to do with xylem histogenesis, or vascular histogenesis, and these sort of inhibition fields. And if your diameter is larger, the arrangement of your leaf primordia are going to be different, and essentially the canalization of auxin in those areas in the developing, axial vasculature is sort of going to be spread apart, and you'll be able to develop a pith in certain regards. So it's a little complicated, but it has to do with the development of vascular tissues. Mm-hmm.
And we have another question in here from Matt asking about vasculature, whether it exists in any examples of gametophytes, or are they?
Yeah, that's a good one. Yeah, so I think yeah, there's like a couple idioblasts. You'll get like, one like tracheid that like one tiny little trachea would be called like an idioblast that will develop in a gametophyte. But it's kind of rare and not very common. Mm-hmm.
All right, and then another good question here from Art about whether you see any major reversals in the complexity of vasculature.
Absolutely.
Over the sort of macroevolutionary scales.
Yeah, so what I didn't show, I guess I didn't explain, but I have this massive reconstruction, ancestral character state reconstruction of vascular architecture, which I didn't get into today. But yeah, you definitely have some reversions in a variety of lineages. You have some reversions in the tree ferns from like more complicated, like polycyclic system to a, simple selenosteel system, which is just essentially a ring of vasculature. You have some reversions in the Schiziales and a variety of different lineages. It's a very case by case basis. And if did Art ask that question? If Art's interested, the gramatids from the perforated dictyosteel, this really complex dissection of bundles, you actually kind of lose that in some of the grammatid ferns, which he'll know, of course, what those are. So.
All right. So we're almost out of time here, or not out of time, but it's about 8:30. There's one last question I'll pose here. Wondering if you see any parallels among climbing plants. There's a lot of interesting vasculature that goes on in some angiosperm vines. Mm-hmm. And there's a couple ferns that are climbing or viney as well. Any unusual patterns in those groups?
Yeah, for sure. So one thing that, well, perhaps the question is, I guess maybe they're talking about some of these like dorsiventral patterns, which we didn't really pick up in the quantitative analysis, but there is that could have been, just because we didn't have that many species within our quantitative circumscription. We sort of did a dual analysis where we first had to circumscribe architecture with a subset, and then we sort of expanded it out to the 3,000 species. But something that is very interesting is there's an analysis. I think Wilson, there's a paper, Wilson and Knoll, probably 2 decades ago now, where they looked at these Medullosin seed ferns, these extinct species that seed plants that sort of twine up other plants, and they have these massive tracheary elements, so these massive conduits that sort of occupy this morphospace of xylem that doesn't really exist, at least from what they saw. But I have I think that you have some of these climbing fern, these rhizome climbing tropical ferns, Polybotria and Lomeriopsis, things like that. And I have a hunch that those climbing species, if they don't have vessels, which vessels have evolved in some ferns, I think they might occupy very similar morphospace in terms of their conduit dimensions to these like Medullosin seed ferns, and perhaps to lianas, which we know have massive conduits as well.
Yeah. All right. Great. Well, thank you for coming out tonight, and thank you to all 3 of our presenters this evening. It's been great to see some examples of the work that you folks are doing, and that's some supported in a small way by the NEBC awards from 2020. It's great to have a chance to sort of check in at this point. It's been a challenging couple of years here, but it really, the work you're all doing is really impressive. So it's a wonderful thing to do, to get together on a January night and see greenery, think about plants, and hear about this research.